We are live, Waldeyr.
Let's go. Good afternoon. We are here to start our technical session number 5. This is the Brazilian Symposium on Bioinformatics, the 2020 edition, for short BSB 2020. And today we're going to have presentation of 3 full papers and 3 short papers. And I would like to invite the presenter of the first paper, Janaina Fortirer, which will present the paper Importance of Meta-Analysis in Studies Involving Plant Responses to Climate Change in Brazil. Janaina, it's up to you.
Hi. I will share my screen. Wait a minute. Okay, can I start?
Yes, please.
Hello, everyone. My name is Janaina. I am a master student in the Bioinformatics program at the University of São Paulo from Brazil. I am here today to talk about the importance of meta-analysis in study involving plant response to climate change in Brazil. My advisors are Marcos Buckeridge and Camila Toledo, with collaboration of Adriana Grandis. Let's move on to introduction. It's clear that is too much information and a lot of scientific data have been discovered at a higher velocity. However, the question is, how do you use this? And moreover, how to apply to life-related problem? 100 papers were published every day. About 39 billion papers were published in scientific journal. The number of papers increased exponentially, and there is a need for revision that compile data production to guide the several study of this area. So the question is how to synthesize the state of art. One way is doing to collaborative work like Intergovernmental Panel on Climate Change, IPCC report that synthesized approximately 6,000 papers about climate change. 91 research from different countries composed this super collaborative report. Another way is to do a quantitative synthesis from individual studies. A highlight of the quantitative synthesis is meta-analysis, which is the statistical methods compares the results of different studies that address common questions. So the meta-analysis can provide more precise results than individual studies. Now, I would like to discuss here the importance of meta-analysis and why do a meta-analysis. First of all, the statistical method employed in meta-analysis can increase power and precision results. Second, it's possible to detect the percentage of the effect and show the statistical difference among treatments with a confidence interval. It's possible to quantify the effect size. Furthermore, this analysis reduces problem with data interpretation for the sample variation. It's necessary to check homogeneity or heterogeneity of results. Answer answer question not driven by individual studies, factors that differ across the study. Finally, when the meta-analysis is concluded, it's possible to check the hypothesis and create others. The meta-analysis can have advantages to synthesis and integrate the difference of studies. However, you may be questioning yourself, when did the meta-analysis come about and when it started to be used? To solve this curiosity, I will give more information on meta-analysis brief history. In 1904, Carl Pearson began his studies with statistical analysis. Fisher and Cochrane did studies with experiments with agriculture, and in 1940, the psychology area synthesized 60 years. So, in the 1976, Glass used the first term meta-analysis. In the same decade, medicine and sociology were the first area to begin use meta-analysis. However, only in 1990, the biology areas has been used in the meta-analysis. Last year, one paper from Buckeridge was published in Nature Journal, say that the meta-analysis is grandmother of big data and open science movement. In biology, the most critical question is the consequence of climate change on biodiversity. Climate change is aggravated by deforestation, such as change in land usage, burning fossil fuels, which increases greenhouse emission like CO2. However, human activity increases the quantity of greenhouse gas emissions in our atmosphere. For example, fossil fuels are responsible for 87% for green gas emission to atmosphere. In Brazil, the most emission result from deforestation. On the other hand, the plants do the CO2 assimilation by photosynthesis. Furthermore, the ocean is another sink of CO2. However, 44% of CO2 stay in the atmosphere. The IPCC uses scientific data to create models that show prediction climate change consequences on biodiversity. The graph in the left shows the relationship between temperature, showing the blue, and CO2 emission, showing the green. And the reduction of CO2 emission is essential to mitigate global warming. However, these works does not contemplate tropical areas because due to the lack studies. The graph on the right shows that the number of the work in temperate biomes is higher than tropical biomes. Therefore, more study is in this area I needed. Okay. Brazil has a high biodiversity with different biomes. Besides the tropical forest can storage tons of CO2. Brazil has a different biology approach as ethanol and biodiesel. However, the big problem is the change in the land use caused by deforestation. So, this study objective was to do a systematic review and meta-analysis in climate change studies to provide one overview to Brazilian research and guide future studies. Let's move to methodology. There are six steps for the scientific method for data analysis. First, we need to identify the problem, which is climate change. Then we need to establish a question to solve. In this case, how many have studied climate change in Brazil? After, make a hypothesis, like there are less Brazilian studies to explain the plant response to climate change on tropical vegetation, and to test the hypothesis by meta-analysis based on systematic review. To do a systematic review is needed the literature search with well-defined criteria. This study search for the effect elevated CO2 on plant at Web of Science, Cielo, and Brazilian Digital Library were used digital library of thesis and dissertation. There were used keywords to identify two studies. For example, this image illustrated the keywords input on the database for search. The next step is the inclusion criteria. In this study, the criteria were study conducted in Brazil with elevated CO2 on plants' physiological response, experiment with FACE or OTC system, and study with statistical data. To do a meta-analysis, we followed this workflow. I would like to highlight the effect size calculation, which promotes the standardization of the work, and the moderate test used data to verify if trees are more responsive to climate change than herbaceous plants. All analyses were conducted in metaphor package in our program. Moving on to results and discussion. This slide summarizes world history events hanging from meeting to IPCC, report that addresses global climate change. And I would like to highlight that Brazil hosted two events, Rio 92 and Rio +20. We found that these political events that analyze global warming correlated with increased in publication. For example, after 44 IPCC events that concluded the human activity increased CO2, we also saw increased in this publication topic. This is showing the black bars, okay? Another example was the Copenhagen conference that talked about the increase in temperature, shows in the red bars, that also increased the number of the publication. Here is a flow chart show the systematic review step. We found more 1,000 studies. However, 44% were excluded because they were not conducted in Brazil or with not experiment FACE or OTC, or did not have statistical data. Finally, the comprehensive studies included in the meta-analysis were 36. However, 60% of these studies were published only in thesis or dissertation in Portuguese language. So it's difficult for people to access the results. This graph, the most variable found was biomass. Biomass measurement due to facility on the method and the low cost. On the other hand, the smaller number of observation in study measure photosynthesis by looking at more complex levels and high cost equipment. Our data show the diversity of variables that should be taken into account in the future for meta-analytical studies of plants in elevated CO2, particularly in study on plant species in Brazilian biomes. Here is a typical graph to meta-analysis called forest plot. Okay, this axis here is the effect size in log response, and the y-axis represents growth with biomasses categories of plants. In the total observation, in the parrots, we can see the mean estimative with confidence interval, and the zero line represents no effect, so that values do not overlap to zero have a significant difference. In this forest plot, we can see that growth variables are representative, and all categories such as tree and herbs. Okay. However, when the meta-analysis was done with photosynthesis variable for ABC4, were the less representative, with only 3 observations, which make the confidence interval large and increase the uncertainty mean estimative. These results show the importance of the high number of observation to do a meta-analysis. This study shows that number of variables measured in Brazilian research that afford meta-analytical approaches is relatively small. The data represented in the yellow boxes are more frequent than the red box. On the other hand, the red box represent the data that need more evaluation. Therefore, for future studies, these variables need to be included to fulfill plant systematic general. Okay? To conclude, despite having too much information, there is also a lack of data on tropical biodiversity. We highlight that more than 60% of Brazilian studies were not published in the English language. This characteristic makes it difficult to assess the Brazilian results by international research. Consequently, the IPCC report with projection model to evaluate climate change in the world will not include the key biomes in tropical climate. Given the importance of Brazilian biomes, agriculture of the planet, and land use for the impact of climate change, we conclude that it is urgently needed that more studies would be performed with tropical species. So thank you for your attention.
Thank you, Janaina. Very nice
Thank
talk. So now I have some time for questions. If you want, any one of you want to make a question, you can just raise your hand or type it on the chat. I wait for a few seconds. Okay. Well, it seems that well, it seems not. It seems I'm certain that this is a very important work, and it is a hot topic because we are all involved in this process of the climate change, and certainly is very important to study the effect of the CO2 in all the living organisms, especially in plants, because we depend a lot of them, right? Also, I agree with you in your presentation that the metadata is very important to establish some kind of support to help in the robustness of the findings, because sometimes it's a little bit difficult to establish the causality of some data and the findings, and the metadata can be somehow helping in this task. And well, you show in your method that you looking for this metadata, you standardized the words, right? Because probably you're gonna find a lot of different ways to say the same thing, in different papers. Most of them are in Portuguese, which means you need to standardize them, maybe translating from a language to another one. Well, this is a very important task that needs to be done very careful. And I would like to know if you have used ontology or something like that to standardize these words before your analysis.
No, in my project, my study, I didn't use ontology because didn't have a genetic study in Brasilia with the effect CO2 on plants. It's a field study.
Mm-hmm.
Genetic.
Yeah, I see, but an ontology can be more general than just for genetic data. It's a kind of a way to standardize how the things can be organized in a hierarchical way. For example, there is an ontology for the names of the parts of the plants.
Yes.
You should say flower or flowers. You should say leaf or leaves. Then I mean an ontology to standardize the words. In this case, could help you somehow to better organize your findings, following already standardized way to call all these words. But probably when you look at that, you will find that you already did it. You just not have seen that you already did it, probably.
In my data, the corpus, the data, it's just numerical data. Your suggestion for the string, the string data, for example.
Yeah, you have your numerical data, right? But when you calculate this numerical data, you calculate them over kind of a class of the metadata, right? For example, growth. You cited it in your presentation. So probably you will find the different words to describe the plant growth in different papers, including
Yes.
Portuguese and English. But you need to read all of them and realize that this word means growth, even in it's written in a different way. But what I mean is with an ontology, maybe you can translate all these different words that have the same meaning for your
Yes.
categories.
Yes. Okay.
Um, as you already did it. You did it, but you not used well-established a well-established database for saying which could be an ontology for
Okay.
these names. You
I
mean?
understand.
Okay, but it is more a suggestion than a question, but
Okay. Thank you.
it could bring you some more robustness on your method, for say.
Thank you for your
Well,
suggestion.
let me check the chat if you have any questions there. No. Anyone else would like to make a question? No. So I thank you, Janaina, for the
Thank
nice
you.
presentation.
Thank you.
Congratulations on your work, and I wish you success in your work. I would like to invite the next presenter, Diego Mariano, who will present a brief history of bioinformatics told by data visualization. I'm excited to see it.
Can everybody see my screen?
It seems that it's coming, but not yet.
Okay.
Just a black screen.
Oh. One minute, please.
Okay. Yes.
Oh, okay. Now it's working?
Now it's working. Go on.
Oh my God. Oh. I don't know what happened.
Okay, don't worry. Take your time. Just
I'm sorry about that.
don't be. It happens. Diego, I can see your PowerPoint. I think you just need to go to the first slide and pull it in the presentation mode.
No, I think my computer just stopped to work. I don't know why.
Oh, I see.
I don't know what happened. I think everything broke here. Could you pass the video?
I think so. Daniela,
Yeah,
are you
sorry.
there?
Waldeyr? Are you listening?
Yes, I'm listening.
It's better to put the video?
He asked it to.
Okay, just a moment.
So let's use the backup.
Yeah.
Oh, just a moment. Sorry, just a moment. Okay. Can you see the video?
Yes.
Okay.
We can see. Let's.
So, hello everyone. Today I will present the paper A Brief History of Bioinformatics Told by Data Visualization. My name is Diego Mariano and I'm from Federal University of Minas Gerais. So in the last years we have seen the rising of new technologies that are changing the life sciences. And bioinformatics emerged to deal with the data produced by these new technologies. So in this presentation, we aim to tell a brief history of bioinformatics based on exploratory analysis of recent publications in the literature. Well, but first, let's start this presentation talking about the age of bioinformatics. So, different from what most of us think, the bioinformatics did not start with DNA analysis. The first computational studies were focused on proteins. Since 1950, experimental approaches have based on exploratory analysis of small protein structures as insulin. And Margaret Ihosvany was a pioneer in bioinformatics studies. She proposed the use of mathematical approach for analyzing biological sequences, and she published the first protein database and introduced patterns to represent amino acid sequences such as the three-letter code, and also proposed the PAN matrix for comparing homologous sequences. So in the years 1970 we have the release of the first methods for DNA sequencing. And for example, the Max and Gilbert method or the Sanger sequencing method, but these methods still not could be used in large scale. First, it was necessary a technological evolution. And at the same time, many methods for biological analysis emerged. For example, the Needleman-Wunsch algorithm for sequencing comparison, and the first method for phylogenetic through inference, and later the BLAST tool used for local alignment. But the game changed when strategies were used in combination with computational approaches. So this was possible due to the improvement in the computational processing power, and mainly due to the reduction of microchip components. And in addition to the popularization of the internet allowed that anyone could connect to the World Wide Web and easily share data. So this evolution also came for laboratories equipment. In 1986, we have the first automatic sequencer using the Sanger's method. So this motivates the scientists to go beyond. And why not to discover the human genome? And the Human Genome was an international, the Human Genome Project was an international research effort to map all of the genes and sequence of the human genome. However, in the middle of the 19 years, the sequencing of the human genome culminated in a race between the public and private initiatives. And due to the more powerful computers, it was possible to use new strategies to sequence genomes, such as the whole genome sequencing strategy, where many copies of the DNA are fragmented in random positions, read by the new sequencer machines, and then the fragments are ordered using assembly algorithms. And this provided a high improvement in sequencing. Maybe we could affirm that this was one of the most important steps for bioinformatics to become what is today. So in the following years, these strategies was used to sequence the first bacterial genome, and in 2001 the draft of the human genome was published. So nowadays we have many high-performance sequencing platforms which can lead to new scientific discoveries and with low cost. So all these evolutions of technological resources has generated a large amount of data, and thus bioinformatics went from just a tool for biological analysis to a consolidated research field focused on the development of new models, algorithms, tools, and new types of analysis to deal with biological data. So consequently, an unprecedented amount of studies using bioinformatics approach have been released, increasing the peer-reviewed published papers. And also many scientific journals focused on bioinformatics studies were created. So in this presentation we used exploratory analysis from metadata collected from the bioinformatics literature to construct visualizations to analyze the state of the art and get insights into the main topics in bioinformatics research. So we hope that these visualizations they can provide insights to understand the recent changes in evolutions and bioinformatics field. So first we collected abstracts and other metadata from papers published from 1998 to 2019 in 4 leading bioinformatics journals: Oxford Bioinformatics, BMC Bioinformatics, Briefs in Bioinformatics, and PLOS Computational Biology. And we used the PubMed API to collect data, and Elsevier API was used to collect citations, and data were processed using Python scripts. And we also constructed a user-friendly web tool using these technologies. And here we have the results. So first, here we can see a list of papers analyzed, and we collected metadata from more than 30,000 papers. And here we can see a general vision of the collected data. The amount constantly increased during the years. And on the right side we can see a first interesting pattern. We have an increase in the number of international collaborations. That means papers with authors from different countries. So here we can see the distribution of publications grouped by regions. We can see an increment in the number of papers published by Chinese authors and a reduction of the United Kingdom proportion in global production. And unfortunately, Latin America has allowed participation in global scientific production. However, Brazil leads the bioinformatics scientific production in the region, being responsible for most of half of Latin American publications. So we also analyzed the keywords used in the papers. Here we have a ranking of the 5 most used keywords in papers, humans, computational biology, algorithms, and so on. But this graph does not expand so much because the most used keywords were really expected. So we try a new strategy to analyze the keywords used. Because of the high number of keywords, we used word clouds to detect interesting patterns, variations across the years. For example, here we have the word humans. Humans appears with a huge size in the 2010-2019 word cloud, but the word is small in other years. So this means that this keyword is most used recently. So this analysis could help us to find interesting patterns. So, for example, we analyzed the most cited omics. Genomics studies are the most cited during all periods, and transcriptomes have increased, and proteomics studies were very cited in the past, but now had a proportional reduction. And on the right side, we analyzed keywords very used in the past, but not used nowadays. So we asked why. So we supposed that it's because they are now trivial. For example, in the beginning of bioinformatics, it was really important to discuss in the literature what programming language should be used in bioinformatics applications. But now we have well established it, and it's not necessary to highlight the language used. So, and talking about programming language, our analysis also show that Python, Perl, Java, and R are the most popular programming language for bioinformatics. Here we highlight that there is not a best programming language. The choice of a programming language depends on the context and your previous experiences, but the existence of biological data analysis libraries appear to be a good motive for using Python, R, Perl, and so on. So we also analyzed the citations of molecular dynamics. Molecular dynamics uses force fields to simulate molecules' behavior during determined conditions and periods, and so it's a method that requires a high computational cost. And we can see in the graph A that the number of studies using molecular dynamics have increased in the last years, and we have correlated this information with the evolution of the graphic cards, because many molecular dynamics use GPU to process data. So in this year we have seen a big increment in the number of transistors present in the GPUs, and a reduction in the price of the graphic cards that could be directly responsible for a possible popularization. And based on this, we predicted a promising future for molecular dynamics and an increase in the number of studies using this. So, and here we have some curiosities. Artificial intelligence is studied since the years 1970, and our analysis showed that it was very cited in the beginning of the 2000 years, but now is few cited. This is really interesting because in 2001, Steven Spielberg released the popular movie AI, Artificial Intelligence, and this may suggest that in some way the pop culture could influence the way that scientists describe their work. So we can see that after a time the use of this keyword reduced, and other specific keywords related to artificial intelligence started to be used, such as machine learning or data mining or so on. And unfortunately, we had some limitations at performing this study, and that we should discuss about. So the main limitations were the name disaggregation and the inconsistency in the databases, mainly in PubMed. Here we have some examples showing that is a complex task to determine if these names correspond to the same person, and here we have a variation of a same affiliation. Okay, and about the databases, we find several problems, missing data, errors. For example, affiliation of the first author is attributed to other authors, and citations calculated wrong, mainly in PubMed. So we try solving these problems with some strategies, but we believe that we couldn't solve all these problems. So here we also applied some disaggregation rules using special the last author name, their initials and affiliations, and manually included more than 40 rules for detecting affiliations and special regular expressions. And in the last case, we performed manual correction, but we had more than 30,000 papers and more than 100,000 authors listed in our database, so it was not possible to apply it to all database. So I know that many of you must be wondering that the work that I presented here today is a different kind of study. Yes, you're right. So what are the main contributions of this study? So here we reviewed the historical context of bioinformatics. We evaluated the state of art of recent publications based on 4 journals, and in the end we made predictions based on our observation and stimulated the reflection about the future of bioinformatics. So one last thing, we collected a list of authors and ranking them based on their contribution in bioinformatics publications, and we also grouped them by nationality, and to try to answer who are the authors that most contributed in bioinformatics. And recently, when I say recently, I say 2 days ago, we constructed a network of Brazilian authors collaborations, and in this graph each node represents an author that are connected by publications, the black circles here, and color represents the affiliation of the authors. For example, here Professor Dr. Raquel Minardi is connected to Professor Carlos Silveira by this paper published in Bioinformatics in the year of 2012, this hydro-pace paper. Okay, and this graph shows some connections between clusters of different institutions, and I highlight this one that connects Fiocruz, Unicamp, and Federal University of Rio de Janeiro and USP. And these connections can represent collaborations between researchers from different institutions, or even researchers that worked in an institution and now are working in another place, which could mean that they are spreading bioinformatics, what is fantastic. And our final reflection is that we hope to see more clusters connected in this graph in the future. So we can access all these visualizations at this link. If you find any error, you can contact me at this email. And I would like to thank you, the funding agencies, CAPES, FAPEMIG, and CNPQ. I would like to thank my university, UFMG, and my laboratory, LBS. And thank you all.
Thank you, Diego, for the presentation. It's a really nice work. So now I have some time for questions. And again, I will wait for a few seconds if someone would like to type the question in the chat, or maybe raise your hand and talk in the microphone. It's free for.
Yeah, sorry about my
my presentation. My second monitor just broken.
Don't be sorry, Diego. It was a nice
Okay.
presentation, even being recorded.
Okay.
No? Well, Diego, about this data, I was very intrigued, and I think this work brings us more questions than answers.
Yeah.
It's interesting work because of that. I think looking for this graphics and this data you bring to us, I started to make a lot of different questions for myself, and one of them was about the graphics where the artificial intelligence was decreasing while the specific particular areas of artificial intelligence were increasing. So it's clear for me that the people are giving more importance to the method than the and finding some differences between the methods, and then they started to highlight one or other method applied to a particular problem, right?
Yes.
And my question about this is, do you think it would be possible to make a kind of regression to maybe to predict some trend topics for the next years in the bioinformatics studies?
I don't know, because each year we have new technologies that are emerging, and so we don't know what will happen in the future. We don't know if there is a new technology that will change everything. And if you think that we're talking about the sequencers, 10 years ago, we don't have the possibility to construct the sequential genome with a low price. So today, this is very simple. This is very, it's not so expensive than 10 years ago. So it's really hard to predict the future, predict what will happen in the future. We just made a simple prediction about the graphic cards, because the graphic cards, we haven't seen an increment in the number of publications about molecular dynamics, and we haven't seen the great improvement in the graphic card, in the number of transistors, and we have more powerful components. So it's we can think that in the future we have more experiments using molecular dynamics. But about the technologies, the other technologies, we can't say anything. We can predict what will happen.
Yeah, I see, but maybe I will rephrase my question. For example, when you see that machine learning is an increasing curve, but when I look for the curve in machine learning, I do not know related to what are the biological problems that machine learning is related to in that curve, right? So, for example, could you say for me, machine learning is a technology that has been used more for topics A, B, or C in biology, while data science or deep learning or something like that is more used for another different
Yes.
topic. So in this way, could you maybe predict? I don't know if predict is the best word, but to identify some trends or some tendencies for solving the biological problems with specific technologies.
Oh, I understand now. I think you're right. It's possible. Let's think, for example, genomics works that are using machine learning approach or transcriptomics or proteomics, something like that. It's possible to do, and we didn't do this in this work, but we have all the data available in the website, so anyone could access the website and download the data and try to make their prediction or anything else.
I see. Well, we have some comments in the chat. Renata says, nice talk. And what are your perspectives for Brazilian bioinformatics in the coming years? And I will put together the question from George, which is very similar. Do you have plans to extend this study focusing on history of bioinformatics on Brazil? Maybe it will be possible to explore more source of data.
Okay, we'll start with the first question was Renata.
Yeah.
Renata?
Renata.
Yeah. So a great question, Renata. I have read a paper last days ago about the future of bioinformatics, and a person had said that bioinformatics will not exist in the future because everyone uses computers for everything. So there is no bioinformatics. There is biology, and people will use computers because computers will be part of everything. So I think that it's not like that, but the future is complex task to predict. But I think that more in the future, more biologists will use computational approach. So this will be really part of biology. And about the second question of George, he asked about the history of bioinformatics in Brazil. Yes, this was the main plan of this research, but we have some problems in this process and in to execute this analysis. And you have seen that I have included a graph of collaborations in Brazil, and we finished this graph just 1 week ago. And we have plans to make more analysis and correlate more the data and understand better how are the collaborations in Brazil between researchers in Brazil. And we hope that this data could be used for I don't know, incentivize people to collaborate more, you know.
Yeah, I see. Well, very nice. I just gonna read some more comments. Gustavo says, I really enjoyed, congrats. Cristal says, excellent work. Jesus say, very good. Danielle, very interesting work. So I have just a suggestion for ending this presentation is in the last graph you presented, the Brazilian collaborations. I think you should include some geographic data to construct some kind of different graphics showing in the map where are the collaborations. I believe would be nice.
Yeah, thank you for the start.
Okay, Renata also says, yeah, I think bioinformatics without biology is just a tool. Well,
no,
that's
we are we are how to say relevant research field. So we have many problems very relevant in bioinformatics to construct algorithms, methods, and other tools. So we are for a while we are really it's not just a tool. I think it's my opinion.
Okay, thank you, Diego. So
thank you.
Let's move on to the next presentation, which will be performed by myself. Let me share the screen. Okay, tell me you if you can see, because now I cannot see you.
Yeah, that's okay, Gabriel.
Cool. Cool. Let's go. So, good afternoon again. I'm Waldeyr, and I'm going to present this work. The title is Computational Simulations for Cyclizations Catalyzed by Plant Monoterpene Synthesis, which was done in collaboration with Daniela, my student, Dr. Jakob Andersen from University of Southern Denmark, Dr. Maria Emilia and Marcelo Brigido from UNB, Dr. Peter Stadler from University of Leipzig, and Dr. Christoph Flan from University of Vienna. So this presentation, we follow this outline, introduction, and then where I'm gonna try to make you have an overview about metabolic pathways and about terpenes, and followed by the methods and our results. Okay, metabolic pathways collectively define the biochemical repertoire of an organism, exposing the steps of its production. And in silico, metabolic pathways have been constructed using a wide range of computational methods, and they can vary in some aspects, among which the data structure and the granularity of biochemical details. And this is this granularity of biochemical details is the key point in my work, which uses graph grammar rules to infer chemical reactions. This is a method that exposes the initial intermediates and final products. So, for example, talking about the granularity of biochemical details, in this example, the classic database of metabolic pathways like KEGG, the reaction details are hidden in behind these arrows, because they show only the initial and the final products. But there is a plenty of steps of intermediate compounds between the initial and the final ones. And in this case, for example, we have here the geranyl diphosphate, which loses the diphosphate here, and after all these steps, you're gonna have this compound, which is a beta-pinene. If you don't know what is it, a beta-pinene is a compound that very used to do the cleaning products. For example, if you have pinosol in your house, probably the smell of pinosol is a beta-pinene. So all these details in these reactions are missing in the most of classic database. So when you look for this reaction, maybe you can lose all this information. Well, in this case, looking for the terpenes, they are the oldest and various plant natural products. They are often present in essential oils. Some of these terpenes, especially the small ones, are volatile compounds that play a crucial role in response to herbivorous and interaction with other plants and attracting pollinators. For example, smell of a flower could attract a bee or a bird, or a smell of fruit could attract birds to eat the fruit and then release the seed. So there is a plenty of ecological roles which are played by these terpenes. Also, they can be used by us, by humans, as flavors in agricultural and industrial products, as fragrances in foods and cosmetics, pharmaceutical stuff, and biofuels. The production of terpenes can be compartmentalized inside the cell. In plants, particularly, the compartmentalization is summarized in this figure, which shows that we have two precursor molecules, IPP and DMIPP, which means isopentenyl diphosphate and dimethylallyl diphosphate. One is allelic isomer of the other. And these two molecules can be produced both by mevalonate pathway in the cytosol and by merytyl erythritol pathway when inside a plastid like a chloroplast. From these two molecules, which are the precursors for all the terpenes, we can have terpenes which are classified according to the number of carbons in their skeleton as hemiterpenes, monoterpenes, diterpenes, carotenoids, sesquiterpenoids, or triterpenoids. Terpenoids is about something like terpene, because inside the cells, terpenes can change a little bit. And well, in plants, we have this situation here, where after you have DMA, PP, and IPP precursors, you can dimerize them. They have 5 carbons, and then you can join 2 of them and have a molecule with 10 carbons, which is geranyl diphosphate. And this geranyl diphosphate is what we're gonna be the precursor for all the monoterpenoids. So monoterpenoids or monoterpenes are those terpenes that have 10 carbons in their skeleton, right? Also, we can have terpenes that are acyclic. For example, this one, citronellol, linalol, geraniol, and we have some terpenes that can have cycles, alpha-pinene, camphor, limonene, menthol, and something like that. This one, for example, linalol, if you look in your shampoo, I bet you're gonna find this one there. And between the components, it will be the flavor of your shampoo. Well, all these molecules are formed by action of an enzyme called terpene synthase, or for short TPS. And this enzyme, TPS, is responsible for taking this precursor, the geranyl diphosphate, and a single enzyme like this one can produce a plant of different and various monoterpenes like this. So these enzymes are called promiscuous enzymes. This means that, as I said, a single enzyme can lead a variety of terpenes in response to environment conditions. Well, this is a problem because when you try to annotate a sequence, it will be not very accurate because the product of the enzyme, the product of this enzyme, the TPS, is not a single product, right? So, well, however, it seems that the terpene synthase have a particular set of chemical mechanisms. And in this work, we took from the scientific literature those mechanisms and abstracted them to simulate the reactions catalyzed by terpene synthase from to form monoterpenes. So how we did it? This is our method. In a top-down approach, what we did was we model the chemical reactions on a mechanistic level, building pathways assembled as a chemical network. This network is abstracted as a directed hypergraph where the vertices of this hypergraph correspond to molecules and the hyperedges to reactions. Each vertex of this hypergraph, representing a molecule, is abstracted by an undirected graph where the atoms are vertices and the bonds are edges. How so? In this example, you can see that you have an undirected graph. It's a graph without a direction in the edge. And you have vertex 1 and vertex 2 and an edge which link these two vertices. And if you label these vertices as with letters representing the atoms, then you have a carbon here and a hydrogen here and a bond, a single bond between a carbon and a hydrogen. Then you can model molecules like this. This one is the same molecule from the example, is a geranyl diphosphate. And when you have double bonds, you have two edges between the vertices. And this is how our approach modeled the molecules. Then, well, if you can transform a graph in another graph, in somehow, what you are doing, if this graph represents a molecule, is transforming a molecule in another molecule. So this is a reaction. For example, if you have a carbon bonded to a hydrogen, then you can break this bond and you have a carbocation and a hydrogen here. And this is modeled using a GML language, a graph model language, like this one. Then we have a software which was developed by one of the collaborators, the Jakob Andersen. The name of this software, I will be never able to pronounce it. It's Danish, but I guess it's Melo-Minardi, which is a software package for chemically inspired graph transformation that can target a set of initial molecules, and these molecules are undirected graphs, and generate the reaction networks automatically, applying a rule-based graph transformation. Well, a rule have a form P, like you can see, where the big L is the left graph and the big R is the right graph, and the big K is the context graph. There are morphisms little l and little r connecting big K to big L and big K to big R. It's called the double push out because it ensures that a reaction can be reversed, which is very common in the nature. That's why it's not only in one direction. And to apply a rule in a graph G, it is mandatory that the big L matches the graph G. In this case, a new morphism M will be applied to entire molecule transforming it. Let's see an example to try to clarify this. For example, here we have a rule in the boxes, in the up boxes, and where you have a molecule of water, H-O-H, H2O is a molecule of water, and you have a cation, carbocation here, C+. Then you have the context, which represents here in purple which atoms should be addressed to match, and then in the other box how the graph should like after the transformation. So you're gonna transform the graph L, big L, and a graph big R using the graph context to find the match. So if you apply this rule into a molecule like this one, if you have a match of this L in this graph G, in this case we have a match, we have a molecule of water here, and we have in some place in this other molecule a carbocation here, then you can use this 2 point as anchor to make the transformation of the molecule in the graph G in another graph H. This is how we model each one of the reactions. In this case, this reaction is called a quenching by water, and the molecule of water will interact with the cation, C+, forming a single bond, and releasing the hydrogen cation. The software will provide the simulator for this rule, and a molecule of water and another molecule of carbocation C containing a carbocation C+, will be given to this simulation. Then we will have a match. We will have a match, and this reaction will take place. And when you do this repeatedly, then you have a very big graph like this example, because you can do it over and over again and in n iterations. And as a reaction over a single molecule can generate more than one result, we abstracted this chemical network as a hypergraph to capture this situation adequately, because we could not have well, what happens here is inside this vertex in the hypergraph, we have another graph, which is a molecule. So the edge is splitted here in two different compounds resulting from the break of the diphosphate in this case. Well, let's go to the results. So we built 17 chemical graph transformation rows, specifically for geranyl diphosphate cyclizations that we found in the literature, and these rules can reproduce using this simulator the blend of monoterpenes that can be produced by plants, and this simulation are able to produce all the cyclization steps in details, explicitly exposing the initial, all the intermediates, and the final compounds, forming a extensive chemical network like the previous picture. Well, taking again the example I showed before, here we have a loss of diphosphate, then we have isomerization of this molecule, then we have ring closure when you bond the carbon 1 to carbon 6. The carbons are numbered beginning from this point, 1, 2, 3, 4, 5, 6. So you're gonna bond the carbon 1 to carbon 6 here, and then you create a ring, and then you have another point to be closed, a new ring from the carbon 2, which is this point, to carbon 7, which is this point that currently is a carbocation. Then the carbocation will migrate to another point, and then hydrogen will be released, and the carbocation will be lost, and the molecule will be stable. And in two ways, could be a alpha terpene when the double bond formed in the first carbon, or the beta pinene when the double bond was formed in the second carbon. So what we did was to create a graph interface, and then you can choose in this graph interface specifically the rules you would like to simulate. In this example, I taken the same rules of this example. We need 3 rules to simulate this isomerization, which are OPP loss alternative, OPP gain by cation, and then 1 to 6 closure, 1 to 6 closure here, and then a 2 to 7 closure, here, and then we need to release hydrogen, which is age loss. And we can do this in 1, 2, 3, 4, 5 steps, right? So we choose here in the iterations number 5 steps. Then clicking in this button, the interface will generate the simulation code that you can use in the software. The simulation code in this case can reconstruct all the reactions, keeping all the details in a data structure that can be computationally assessed. This generated hypergraph here has the beta pinene here in this point, and all the other compounds that can be generated using the same set of rules and the same number of steps. So you can see beta pinene here, and all these other compounds can be generated using the same chemical mechanisms and in the same number of steps, right? As usually in science, we are not alone, and we have some related works to compare. We choose here 4 of them that we consider more related, and although they provide comparable results, this approach differs from ours by using distinct methods and data structures to represent the molecules and their transformation. These 2 in the bottom place is retro rules and biotransformers uses chemical reaction descriptions, which are encoded by SMARTs and SMIRCs, which are approaches that use the SMILES to represent molecules. If you don't know what SMILES is, it's a kind of string to represent a molecule. And they could be compared to our rules, but in a totally different data structure. Then Isegawa and colleagues used a method called artificial force-induced reaction, which is a little bit more elaborated method, but it takes a very it's computationally resource consuming, for saying. And in terms of results, the work from Boxui Tian, as well as our work, can generate all the possible carbocations and monoterpene synthase, defining and organizing the product chemical space from basic molecule skeletons. So basically, they say they can do the same thing, but using a different approach. I going to the end of this presentation, just resuming the results. These monoterpene rows were added to the previous results for sesc terpenes, which are terpenes with 15 carbons. And they are composing a terpene biosynthesis simulation system called 2PEF. It brings, and this time made by my student Daniela, a web interface that makes the writing simulation more intuitive and less laborious, as some drag and drop and clicks are enough to generate your simulation code, which facilitates for the biologists or chemists. And the work is available at my GitHub. You just need to look there, including the interface. You can use it online. Thank you all for paying attention. I would like to thank you, all my colleagues from PSP2. I'm gonna stop share to see you and see the questions. Okay. Oh, Danielle. Danielle says, nice work, Waldeyr, thank you. How do you manage the provenance of the analysis? Do you store the chosen parameters, such as number of iterations that the user set to generate each simulation code? Is it important for the analysis? Nice question. And we did it in the previous work when we were looking for sesc terpenes. We started with sesc terpenes, and now we are expanding the row set with monoterpenes. And in the previous work, we used a graph database to store the provenance, but the provenance is the hypergraph for itself. That's why we use a graph database, because of the transformation to store the network was minimal. We just take it almost like it is and stored in a graph database. In this case, we tried two different graph database. We used Neo4j and we used OrientDB2. But at the end, we choose it Neo4j because it's a little bit easier to write the queries, in my opinion.
Thank you, Waldeyr.
Yeah, there is another comment here from Gustavo. Can you also use this approach to study terpenes in bacteria, or the database is focused on plant production? Well, Gustavo, the rules that we wrote were plant inspired, but there is a lot of intersection between the mechanism that are used by bacteria and fungus too, with the plant ones. And I just choose plants now because I need to stay inside in a scope. But we have plans to in a future use the expand also for those rules that are particular for fungi and for bacteria. Did I answer well? Is it clear? Okay. Okay, thank you. So let's move on. I take too time from you. Sorry for that. Cristal, is up to you. Cristal will be present the work oncogenic signaling pathways in mucopolysaccharidose. My God, I'm so sorry.
Mucopolysaccharidoses.
Thank you very much. It's more difficult than the methylphosphate.
It's the biggest word and scare word.
Go on.
So, can you see my screen? Can you hear me?
Yes, I can see it and I can hear.
Let's begin. Also, and hi, folks. When Waldeyr says my name is Crystal, today I present my work entitled Oncogenic Silent Pathways in Mucopolysaccharidosis. It's a pleasure to be here in this meeting. And I'm PD student in PPGBM in the Federal University of Rio Grande do Sul. And this work have shared with my colleague Luis Dias and my advisor Ursula Mate. So this is my agenda of this presentation. And I first talk a little bit of the relation between cancer, lysosomes, and glycosaminoglycans. Today I say gags. It's more simple. And so cancer cells depends of several organelles, such as the lysosome. The lysosome are responsible for the degradation of macromolecules, but are several functions that means the lysosome too, such as gene regulation, apoptosis, antigen presentation, and in cancer, the lysosome also participates of tumor invasion and metastasis. So the lysosome contains several hydrolyses, such as enzymes, and the defects in the lysosomal enzymes culminate in the lysosomal accumulation of macromolecules or metabolites, like the glycosaminoglycans. So these gags are present in the extracellular matrix, and the gags interact with a wide range of proteins and receptors. So these defects in the lysosomal enzymes of these genes culminate in the mucopolysaccharidosis, which means a rare group of genetic disease, when the deficiency in lysosomal enzymes are responsible for the worst degradation of these gags. And these patients present several progressive multisystemic involvement and in several organs and tissues like cardiac tissues and bone tissues and several central nervous system impairments. There are 11 types of MPS, which vary according to the enzyme defects and the accumulated gags, such as heparan sulfate, dermatan sulfate, and so on. So, in MPS, secondary cell disturbance affects pathways common to cancer. For this reason, these aims of this work were to identify oncogenic pathways related to cancer in the different datasets of MPS available in public databases like ArrayExpress and Gene Expression Omnibus, and to compare the ontologies across the different types of MPS. So we here see these methods of these works. We have one dataset of dog, 3 of human, 8 of mouse. These datasets are from MPS 1, MPS 2, MPS 3A, MPS B, MPS 6, and MPS 7. So we perform the gene expression in our environment with several packages such as EDR, Lima, and we use different packages according with the used platform of the experiment. So the enrichment analysis was performed in Cytoscape plugins like Bingo and Clugo and Clupedia. And to reduce the redundant terms, we use QuickGo database. For more information about these datasets, please visit our 2MPS base in this address. So the summary of the results of this work, we have 12 datasets. We found 1,440 ontologies. And when you use the redundant analysis, we found 680 oncogenic ontologies across the KEGG pathways, biological process, molecular function, and cellular component. We have ontology that appears in all the datasets like hyposinally pathway and mapinase sinally pathway, both of them part of KEGG pathways. We found ontologies that appears in 11 of 12 datasets like proteoglycans in cancer, RAP1 sinally pathway, and cytosine mediate sinally pathway. These two were presenting KEGG pathways, and this one, the geobiological process. And we have ontologies that appears in all of the 6 MPS types analyzed, and we have 11 pathways, all of them of the KEGG pathways. So we have prevalent oncogenic enriched pathways of the datasets analyzed. We see here a plot of all of the 12 datasets analyzed. And we have all ontologies, KEGG, biological process, cellular component, molecular function. And the dataset with the most enriched pathways are this one of the MPS type 3B. And here we see a dot plot with the most abundant ontologies, and we see the frequency, which means 6. It's a ontology which appears in all of the 6 datasets, and so on. When we know that glycosaminoglycans or GEGs play an essential role in the composition of the extracellular matrix, and the GEGs interacts with a wide range of receptors and proteins, and these GEGs helping to regulate processes such as proliferation, cell adhesion, metabolic signaling, apoptosis, cell migration, antigen presentation, and so on. And these processes are altered both in cancer and in the MPS disease. So we only for illustrate, we have adaptation of the KEGG pathway proteoglycans in cancer. We see the heparan sulfate, a type of GEG, And we see several pathways like AKT, mTOR, signal pathway, AGFR, and apoptosis, silent pathway, Wnt silent pathway, focal adhesion, which appears in all of the datasets analyzed. We so conclude with the glycosaminoglycans and participates of several processes, which is essential for the cells and to both in cancer and in PS. So our conclusions were studying the tumor ontology signature in lysosomal disorders may help us understand lysosomal storage disease and cancer's common underlying mechanism. This may help amplify the therapeutic approach for both of types of the disease. And I want to thank the organizers and the persons with here in this meeting, and I want to thank the financial support of CNPq, Hospital de Clínicas, CAPES, and for more information about the MPS base, please scan this QR code, and I open for the questions. Thanks.
Thank you, Cristal. So, as usual, if you had some question, you can type it on the chat or open your microphone, which is free for questions now. I wait for a few seconds.
Thanks, Luis.
Yes, Luis always says good work. I agree.
I agree so much.
Yeah. Okay, I just have one question, and I have been working with some networks too, as you said before, but I have never used Cytoscape, and I see that a lot of works here today cited Cytoscape to analyze their network somehow, and you have used Cytoscape to generate through some plugins the gene ontology analysis, right?
Right.
And I saw that you use different plugins for the same achievement.
Yes.
All of them are from gene ontology analysis. And how is done how do you merge these results from different plugins? Is it a feature from Cytoscape, or do you have to do it by yourself?
And Cytoscape generate a lot of networks, such as one for the bingo analysis. We've use the gene ontology, biological process, molecular function, so on. And for the CluGo, we have used KEGG pathways, Reactome, and other database. We can save the network as a table, CSV, and we plot some for air environment in this one. And for the reduce the terms, which means, for example, we have a term entitled regulation of apoptosis in one database, in one dataset, sorry. And in other dataset, we have negative regulation of apoptosis. So to compare these terms, we use the QuickGo database. We have a plot like an ancestor chart, which contains a wide range of terms, and these terms are the mother term, and we reduce these terms for to compare better the results.
I see. Yeah. Nice. We have some comments in the chat. Raquel says, excellent presentation, Crystal.
Thanks, Professor.
Jorge Kutiji says, nice work, Crystal. Have you used other source of pathways or just KEGG? As suggestion, he says you can see pathways from Reactome, but you already say that.
Yes, Klugo have KEGG, Reactome, Interpro, weak pathways, a lot of resources for ontologies.
Very well. Well, we have new congratulations here from Renata and again from George Buttigieg.
Thanks, guys. Thanks.
Well, thank you also. Let's move on to the next presentation. So now the presentation is the presenter will be Jesus, right? Jesus, are you there? Okay, Jesus will present the natural products as potential inhibitors for SARS-CoV-2 papain-like protease, an in silico study. So I believe you have the power to share your screen. Okay, just unmute your microphone and you can start when you are ready. We already can see your presentation.
Oh, sorry. Good afternoon. Can you see my screen?
Yes, I can see. Would be nice if you click on the button presentar to maximize it for us.
Okay. Okay. Well, hello, everyone. I am Jesus Alvarado. I will present our work, natural products as potential inhibitors for SARS-CoV-2, papain-like protease, and in silico study. This work was carried out with Fabian Jimenez, Gerson Cordova, Ihosvany Camps, and Wilmar Puma-Zamora. Before starting, I want to thank the Brazilian Symposium on Bioinformatics 2020 for the excellent organization and for giving me this opportunity to participate. The outline consists of introduction, methodology, results and discussion, and conclusion. SARS-CoV invades cellular mechanism in this point, in point 1, SARS-CoV-2 entry in point 2. Sorry. Okay. In point 2, membrane fusion and viral RNA release. And 3, translation. And 4, some of these proteins from 3 and RNA from 2 form a replication complex to make more RNA. And 5, SARS-CoV-2 packaging in Golgi apparatus. And in 6, SARS-CoV-2 release. Among these proteins, one of the most studied together with the main protease and the protein spike is the papain-like protease, or PL-Pro, which is necessary to facilitate the spread of the virus and help to evade the host immune response. One of the most outstanding aspects of SARS-CoV-2 PL-Pro is its role as an ubiquitin specific protease, especially of ubiquitin-like protein ISG15, which is a known regulatory agent of human immune response pathway. In this way, it is proposed that blocking SARS-CoV-2 could stop the viral infection and improve the immune response. On the left, we can see the alignment of SARS-CoV-2 with SARS-CoV-PL-Pro structure. The SARS-CoV-2-PL-Pro in cyan and SARS-CoV-2-PL-Pro is in yellow. On the right, we can see the amino acid sequence alignment of PL-Pro from SARS-CoV-2, MERS, and SARS-CoV-2. The conserved residues are in black, and the variable residues are highlighted by a red background color. The catalytic triad is highlighted in light blue. Here, here, and here. The active site residues are enclosed in black rectangles. For example, this or this. Based on this information, the simulation box was defined. Since today, there is no officially an effective vaccine or antiviral against this coronavirus, we highlight the importance of rapid search techniques like virtual screening, or VS, and molecular docking to identify possible inhibitors to this enzyme, using the evaluation of molecular interaction in its active and allosteric site. In this work, we focus on natural products, or NPS, because on these organic molecules, we can find a large surface area and multiple functional groups capable of interacting by hydrogen bonds or van der Waals forces, etc. We carry out two stages. First, virtual screening with Autodoc Bina and 213,038 chemical structure of NPS collected in the freely available Universal Natural Product Database. To ensure the reliability of the BS results, the second stage consists of molecular docking divided in 3 sub-stages with glide from the suite Schrödinger. The best hits of the virtual screening were docked with standard precision, or glide SP, extra precision, or glide XP, and then with the induced fit docking protocol, or IFD protocol, and then with the, that allows flexibility to the residues from the pocket. Finally, to the binding free energy, or Gibbs free energy, of the best 10 complexes from IFD protocol was calculated using the physics-based MMGBSA method with the prime model of the suite Schrödinger. In silico methodology, from the protein data bank, we retrieve the structure of the SARS-CoV-2-PL-Pro with PDB ID 6W9C. For BSA, all water molecules and ions were removed and polar hydrogen were added to the receptor using Autodoc tools. For molecular docking, the protease was prepared by removing unnecessary water molecules and ions with the protein preparation wizard module, and consider the protonation state of the residues at pH 7.4 with EPIC. The structure of NPS in inchy strings were obtained from the UNPD ISDB. 3D coordinates were assigned to them, and then they were geometrically minimized with 20,000 steps using the OpenBabel software. For BS and molecular docking, we add appropriate hydrogen for pH 7.4. For the validation, we use the crystallized structure of SARS-CoV-PL-Pro to check the suitability and accuracy of BINA and GLIDE as a property docking tools. The protein in its native ligand were prepared under the same condition. The box size and the coordinates of the centroid of the binding site were calculated in the same way as for BS and molecular docking stages. Ligands and protein in PDBQT format were used as input files. The centroid of the binding site and the optimal box size, which covered the active and allosteric sites, were obtained with the POCASA server and the eBox site script, respectively. BINA was used with an exhaustiveness level of 24 and the Kinde-1 supercomputer. The best 1,000 ligands were selected from BS according to their binding affinity values. These were docked with GlySP and the SARS-CoV-2 PL-Pro active and allosteric sites. From the results with GlySP, the best 100 hits were selected and docked with GlyXP. From the last step with GlyXP, the best 10 hits were selected and again they were docked with the IFD protocol, allowing flexibility to the residues close to the active and allosteric site. For these steps, the OPLS 2005 force field was used, and the same condition of binding site coordinates and box size from the BS were used. Finally, the Gibbs free energy of the best ligand receptor complexes from IFD was estimated according to equation with the prime model using an implicit solvation model and the OPLS force field. Using the OpenBabel OBRMS tool, the root mean square deviation was calculated between the native co-crystallized ligand of the SARS-CoV PL-Pro and its best redocked conformation obtained with BINA, 0.47 Armstrong, GlySP 0.57 Armstrong, and GlyXP 0.81 Armstrong. Since root mean square deviation values are less than 2 Armstrong with respect to the native ligand, it is considered that the protocols used in the present study are reliable. According to BINA, 88,848 NPS obtain a binding affinity between 6 and 7 kilocalorie per mole, which represent 41% approximately of all ligands. The 1000 NPS select for the following molecular docking test had a binding affinity between 8 to 13 kilocalorie per mole. We identified 10 molecules with the best gift free energy values. Hydrogen bonds or H bonds interaction were found with many residues from the subsite S3 and S4 underlying the table, this or this. These molecules could block the enzyme activity through no covalent interaction with many residues of the SARS-CoV-2 PL-Pro allosteric site, inducing the closure of the loop. These hits are relatively large glycosides and tannins with previously reported biological properties. For example, this is one of the best molecular dockings in green line. This molecule has 9 H bonds and 7 lipophilic interaction. And well, and this study where we identified 10 molecules that can interact favorably with residues of the allosteric site of PL-Pro, being considered stable complexes according to their Gibbs energy-free values. This search was done using a library of 213,038 structure of MPS collected in the UNPD, and techniques like virtual screening and molecular docking. These molecules are mainly glycosides and tannins, and their molecular structures are relatively large, presenting between 6 to 9 H bonds and lipophilic interaction with residues of SARS-CoV-2 PL-Pro allosteric site. However, still are needed prediction of pharmacokinetic properties and molecular dynamic simulation to evaluate the possibility that these molecules can be used for in vitro PL-Pro inhibition assays or lead optimization process. Well, the team gives special thanks to Siembra Empresa Pública in Babura, Ecuador, for their support with Kinde 1 supercomputer. Also, thanks to Dr. Min Hu and Dr. Li Wang of National Supercomputing Center in Jinan, China, for their support with the implementation of BINA, a helpful discussion. And to Dr. Ana Valderrama for her personal financial contribution of the project. Finally, remembering that sin ciencia no hay futuro. Thanks.
Thank you, Jesus.
Thanks.
Very nice talk. As usual, if you have some questions, you can just take the microphone and say, or type in the chat. I wait for a few seconds. Very well. Jesus, I have a question, and it's a very biased question, because I saw in your results you did found terpene glycoside, right? It was one of the molecules that bind. Can you hear me?
Yes.
Yes. Okay. Then I saw in your results that you find a terpene glycoside, right?
Mm-hmm.
Do you know exactly which terpene is that one, or is an unknown terpene?
Yes, I prefer. Posso falar um pouco de português porque o meu inglês
Oh, okay, no problems.
Desculpa. Bom,
no problems. I can speak in Portuguese, and you can answer in Portuguese or in Spanish, and then I can translate again to English. Eu vi que nos seus resultados você encontrou um terpeno também.
Uhum.
Você sabe exatamente que terpeno é esse? Ou é um terpeno desconhecido?
Não, sim, a gente colocou aqui uma tabelinha, uma tabela,
Sim.
com todos eles, né? Por exemplo, aqui o terpeno, ele já foi publicado. O que a gente está fazendo agora é o seguinte passo, é fazer as simulações de dinâmica com eles para reconhecer a estabilidade.
Sim, mas você sabe que terpeno é esse?
Sim, a gente pode identificar pelo ID, pelo código. Essa é a pergunta?
Sim.
Sim, todos eles estão já publicados. A gente tem os testes das caracterizações químicas.
Tá, e esse banco de dados ISDB é de acesso público?
Sim, é de acesso
Muito
público.
Bem. Então eu
procuro. Eu estava levando a resposta por um lado, porque queria comentar aqui que aquele supercomputador que a gente estava usando, ele teve alguns problemas e teve que fechar, problemas da política do Equador. Eu sou de Peru, mas outro problema. Então, a gente está procurando também fazer as dinâmicas.
Sim.
E agora não temos a possibilidade de poder computacional para fazer. Estamos na procura daqueles testes. Acho que é uma boa oportunidade este congresso para conhecer os contatos, né? A gente que trabalha com isso.
Vamos lá, tudo vai se resolver. Obrigado, Jesus. Então, so just for record, and I asked Jesus if he knows exactly which terpene was this one between the results. He says this terpene is already published, and there is and is available in a public database called ISDB, and in his paper there is the ID of this terpene in this database, and we can go there and look for details in these particular results. Also, Jesus have commented that he's looking for scientific partners, especially those who have some supercomputer to continue his work. If you are one of these people, please be in touch with Jesus to make the science go on. Thank you, Jesus. So let's move on. This session should be finished right now, but we had problems with a paper yesterday, and this paper was reallocated to this part of session, and we have the backup presentation today, right? Danielle, is it ready? So we're gonna put this presentation. Unfortunately, like yesterday, the author could not be here to present, but this time he sent his presentation and we play it, and after that we're gonna finish the session.
Okay. Can you see my screen, Waldeyr?
Yes.
Okay.
Hi, my name is Alex. I'm a biological science student on the Federal University of Technology from Paraná, Brazil, and I'm here today to present a paper entitled A Scientometric Overview of Bioinformatic Tools in the Pseudomonas putida Genome Study. So here we have my research group, which helped in the construction of this document. And to begin start off talking about the subject, we need to understand why it's so important to study the genome of Pseudomonas putida. So this organism is an ubiquitous bacteria, very common in soil, and also it is chemoorganotrophic. For this, it is very used in the bioremediation processes, especially when we talk about the degradation of recalcitrant compounds, like pesticides. So we have a lot of documents, a lot of researches using this organism, applying to the processes on environment, especially when we have contaminated environments with recalcitrant compounds. So the objective of this study was to create a global panorama about the publications of the genome analysis of Pseudomonas putida. And for this global panorama, we use the scientometric method. So to create the scientometric analysis, we selected the Clarivate Analytics database, Web of Science, using the terms genome, Pseudomonas putida, and soil. So in the first beginning, we had 159 documents, but we had to do a manual filtering of all the documents because we wanted only the documents who did the sequence of the genome of Pseudomonas putida, and also which have the focus on the bioremediation processes, especially talking about the degradation of recalcitrant compounds. So in the end, we had 29 documents, all of them doing the sequencing of Pseudomonas putida, and all of them we focus on the bioremediation processes. So after that, we extracted the data to Microsoft Excel and Sitespace software to concatenate the data and create the images. So here we can see the number of publications, the number of citations per year, and also the mostly publicating countries. So we can see that the publications started in 2002, and we had a gap of almost 10 years, only having new documents in 2013. And we can see also that the number of citations was almost had an almost constant growth until the current year. The principal countries publicating about the subject was France, Spain, United States, Germany, and Japan. But we also can see that in the current year, we have China with the biggest number of publications. Here, we can see the knowledge areas most frequently in the documents. We can see that microbiology is the most frequent knowledge area, which is coherent because the object of this study was in a bacteria. Also, we can see that environmental science and ecology have a bigger frequency too, because this bacteria is widely used on the environmental processes, like I said before, applied to degradation of recalcitrant compounds. So here we start talking more focused on the genome. We can see that 117 genomes of Pseudomonas putida were sequenced in the 29 documents. It's important to say that in the documents we removed, most of them used the genome removed from databases. So they don't did the sequencing, but they used the sequence removed from databases. So in mostly of the papers, we have the sequencing of the whole genome, but we also have some papers with sequencing draft genome. Also, as usual, we can see that the Illumina platform was the most used for sequencing the genome on these documents. Here, so we can see the assembly software mostly used. SPADIS has the biggest number of uses in this dataset, followed by CLC Genomics, and still 22% of the documents didn't inform in the assembly software. We saw that is very common even in the documents we removed, that most of the documents doesn't inform the software is used on the bioinformatics analysis. Here, to annotation softwares, we also can see that a lot of documents didn't inform the annotation software used on the analysis. But we can see the NCBI prokaryote genome annotation program was the widely used. And again, like I said, here few documents didn't inform the annotation softwares. So Pseudomonas putida is a very studied organism with a lot of applications, especially when you're talking about the bioremediation processes, biotechnologic processes, like the degradation of recalcitrant compounds, especially when you talk about the environmental, because a lot of these compounds are bad for environment. So these organisms are a very interesting alternative to mitigating these contaminating problems on the environment. So for this, the study of the genome of these organisms is extremely relevant because with the molecular information, we can understand more about the metabolic pathways and the genes applied to degradation processes. So this is the most interesting part of the scientometric analysis. So we can see a global panorama about the publications. We can see the countries which have more publications. We can see the focus of the researches, and we can see the status of the researches. We can see the directions of the researches from any subject. So I want to thank the organization of this amazing event, to my research group, my university, my funding agency, and I want to encourage more people to do more scientometric analysis because it can open a lot of doors in the science to understand more what we are researching and to give more informations for future researches. Thank you very much.
Well, so this is the end of this session, and I just would like to, before to go home, I'm already home, but you know, I would like to invite you to our last session, which will be occur tomorrow, on November 27, at 2 p.m. when we're gonna have 3 activities. The first one is the Best Paper Award announcement. Then we're gonna have the talk entitled Searching for Genomic Variants in Multiple Genomes at Once, by Jan Stoy from Bielefeld University. And then some closing remarks. And if you want to know where is going to be the Brazilian Symposium on Bioinformatics 2021, don't miss this session. We're going to reveal. So thank you very much for attending this meeting, and see you.